Assalamu alaikum wa rahmatullahi wa barakatuh. My students, today, inshallah, we begin a new session about respiration. We finished the visit the last time, but we want to talk a video about diseases. This video in front of you now about immunity and disease. But before that, I want to ask, okay, anyone, what is transmissible disease? Anyone want to share with me? Anyone want to start with me? What is transmissible disease? Okay, and what is pathogen? All of the students hear me or no? Please turn on the video. Turn on the video, please. This one. Someone open the video. Now we'll talk about the disease. I want someone to participate with me. Okay, what is pathogen? What is pathogen? Anyone know? Okay, chat. Okay. You hear me?
It's a disease-causing organism.
I don't hear. Please your voice.
It's a disease-causing organism.
Disease what?
It's a disease-causing organism.
Okay, fine. You hear my voice well?
Yeah.
Michael, okay? So that
Yes.
is so we will revise something inshallah in chapter 10. Then chapter 11 and 12 about respiration and inshallah. So now
Okay.
I want to ask another one about what is transmissible disease. Oh no. What is transmissible disease? Amna, yeah. Amna. Who read it hand? Ala al-Sini. Ala. What is transmissible? Ala.
It's a disease.
Who are you, Ala? Okay. What's your transmissible disease? Okay, Ala. What is this? Ala, are you with me? Another one. Amina?
Yes. A transmissible disease which the pathogen can be passed from one host to another.
Thanks. Okay. But a transmissible disease, okay, we'll talk about today because this video is about transmissible disease today. Okay, listen to the video.
There are thousands of different bacteria living on our skin and other surfaces. A pathogen, such as E. coli shown here, is a type of bacteria that causes disease.
So a bacteria type of bacteria, all of the students hear me or not? Okay, Ala, you hear me all? You hear the video all, Ala? You have a question? Ala, raise your hand. You know, the video is okay for you? The video is okay? You hear the video all?
A transmissible disease is one that can be passed from person to person by direct contact, such as touching or through bodily fluids such as saliva. In this experiment, the number of bacterial colonies grown from hands before and after washing will be compared. Agar is a source of nutrients for bacteria to grow on. It is sterile to avoid contaminating the results. By pressing fingers on the agar, bacteria is transferred and can grow to form colonies. A bacterial colony is a visible cluster of bacteria. Each colony can grow from just a single bacterium.
Adhesivity of the leg.
3 fingers are pressed gently onto the agar in one of the Petri dishes. Any bacteria on the fingers should be transferred to the surface of the agar. A lid is placed on the Petri dish. 2 small pieces of adhesive tape are used to seal the lid on 2 sides. The lid should not be sealed all the way round, as oxygen would not be able to get in and dangerous anaerobic bacteria could grow. This Petri dish is labelled as unwashed. Hands are now washed thoroughly using hand wash.
So observe how does she clean her hand, right? So in this day there is corona, no?
Hands are dried thoroughly and the same 3 fingers as before are pressed onto the agar of the second Petri dish. The same fingers being used is a control variable. The Petri dish is sealed on 2 sides using adhesive tape as before, making sure that the seal is not all the way around. The sample is labelled washed. The samples are placed in a sealed container and left for at least 24 hours at room temperature, which is about 21 degrees Celsius. This allows enough growth so that separate colonies can be counted, but not so much that the plate is overcrowded.
The information about activity inside the lab. 25 seconds, the will grow.
After at least 24 hours, the Petri dishes are observed. It is safe to handle and view the Petri dishes as long as the lids are sealed. If a Petri dish is found with the lid off, it should be immediately put in a sealable bag and disposed of in a biohazard bin or an autoclave. Hands should then be washed thoroughly with soap. The number of colonies are counted through the lid. The lid should not be removed as the inside of the Petri dishes are a biohazard. Each bacterial colony that is counted should be separate and have a defined edge. For example, these are some of the common shapes of bacterial colonies. The large patch at the top has an undefined edge and a fluffy appearance, and so is likely to be a fungus. This is not included in the count. 32 bacterial colonies have grown. Some of them are very small and difficult to see on screen, but they are separate and so are counted. Notice this bacterial colony has a very different shape to the others. It is not a fungus because its spiraled edges have a defined outline. It looks like a rhizoid. These 2 also look different to the others. They are not circular, but they do have a defined outline. They look like they are irregular. The different appearance of the colonies indicates that they are the colonies of 3 different types of bacteria: circular, irregular, and rhizoid. The number of colonies grown for the washed hand samples are then counted. It's difficult to see in this image because of the air bubbles under the adhesive tape, but there are no bacterial colonies present. This looks like a fungus as it does not have a defined edge. Although most of the bacteria on the hands are not harmful to healthy individuals, they can be hazardous when grown in colonies. Therefore, it is important to wash the hands thoroughly after handling Petri dishes that contain colonies, or gloves could be worn and disposed of in a biohazard bin. Notice that there are more bacterial colonies growing on the unwashed sample than on the washed sample. There is also a larger variety of bacteria types. This experiment demonstrates that there are bacteria living on our hands that can be transferred by touch, and that washing hands reduces the number of bacteria on the hands. Normally, it is expected to see a few bacterial colonies even after hand washing because naturally occurring bacteria that are healthy for us are released from the skin when it's rubbed during the hand washing motion. This experiment emphasizes the importance of washing hands after using the toilet or before eating to prevent the transmission of bacteria, including pathogens that can cause disease.
This video is very important in this day exactly. Now we'll go to solve some past paper about alternative to practical question. That's paper 6. So we will talk this question first. We are talking about chapter 10, chapter 11, chapter 12. We revise now. We will continue to revise. Figure 2 shows 3 sets of apparatus, A, B, and C. That is A, that's B, that is C. That is C. That is A, that is B, that is C here. Check. Look at it. Well, used to measure different biological processes. For a process, for another process, this for another process. Name the process that can be measured by each apparatus. I want number A, what is the process? Number B, what is the process? Number C, what is the process? Anyone can answer? Just think now. Okay. Okay. On the process now. Then we go to question number 2 about name the gas produced by the voltmeter using apparatus suggest one possible control. That is about what? This is about okay, paper 6. Okay, then we want to go to extension questions. But think in this question now. Think in this question. Think everyone. Who can answer? I want to choose one. Now, who can answer? I want to understand. No one answer? Go to the other question. But do you remember photometer? This photometer, okay, I will show it to you. This photometer, this one. Okay, it depends on transpiration. Remember, photometer depends on rate of transpiration. This process transpiration. If you remember, I will ask you now about what is transpiration now. What is transpiration? What is transpiration? What is transpiration? Who knows? Who knows what transpiration? All of the students with me? Who know what is transpiration? Okay. The students? The rhythm of water in the form of water vapor? Transpiration, this one. Number 1? Okay, my students. the questions about that is about paper 6. I will give you think about it. That's about question. Show part of the lower surface typical dicotyledon leaves. That is the dicotyledon leaf. On figure, label A and the cells B and the cell C. I think that is easy for you. Okay, I will send to the practical inshallah and the homework. Then you will do it by yourselves. You will study well sentence variation. Study well what it is. This question is very important and very easy. This figure label part A and cell B and C. This is about cell inside the typical dicotyledonous leaves. Meaning have it has two seeds. Okay. Who can answer on this question? I want to take part. Who can answer? Do you remember stoma and stomata? Do you remember stoma and stomata? I will give you 2 minutes to think on this question. Just 2 minutes. Then raise your hand. Okay. This question. The surface of the leaf is a figure label part A, part B, part C. Part A, B, part C. Very easy. Who can answer? I'm waiting for you. Just 2 minutes. Who can answer the question? Number A is what? Number B is what? Number C. It's easy. It's about stoma. One can answer. This stoma, this is guard cell. It's about, do you remember lower epidermis? In the leaf, we talk about upper epidermis and lower epidermis. So lower epidermis is about stoma. So now, that is the stoma. The stoma found where? In the lower epidermis with our upper epidermis. I'm asking you. Stoma found in the upper epidermis of the leaf of the lower epidermis of the leaf. You listen to me? Okay. Salman, where is Abdullah? Yurnol. Okay, this is the question. This is the answer of the question. We go to another question here. In figure 3, label part we label the surface of the leaves of key species of plant were studied and the number of stoma per unit area stomata frequency was recorded. Cobalt chloride paper changes color based on sulfur. Piece of cobalt chloride paper were attached to the upper and lower surface of leaves on pot plants. The plants will set up in 1 hour during the day. And color changes were pot plants. The plants were set up at 1 hour during the day. Any color changes were recorded. The experiment was repeated for 1 hour at night. Table 1 shows the result. That is the table 1. Describe the difference in stomata distribution between the two species of plant. Okay, that is in formal order. We have two species, Kezia Visciola and Ruhenia Mondra. That's just to name these two species. During chapter 1 about genus and the general species. Okay, we have two plants. Lower surface is 0, upper surface is 2, lower surface here is about no lower surface, upper surface okay, lower surface no, upper surface that is in the night, that is in the day. That is in the night, that is in the day. But Ruhenia Mandra, lower surface 22, upper surface 0, that is stomata frequency. Okay, we put the stomata here in the lower, more in the lower surface in Bovina combat. Not upper surface, no stomata in the upper surface. Because we have cuticle. Do you remember the real? Okay, I will do just 1 minute. Just 1 minute, okay? 2 seconds. To draw the upper surface and lower surface. The upper surface of the plant of the leaf contains epidermis. This epidermis, okay, contains cuticle here. This cuticle just not prevent it, reduce the water to the water. You don't have parasite cell here. So, parasite cell. You have a parasite cell here. Okay, a parasite cell like that. We have this one here. So we have the lower epidermis, which contain what's called stomata. That is the stomata. Stomata of one, stomata of two. Where is the guard cell? We have the guard cell here. The guard cell, as we said in question number 1. Okay. With the guard cell, that is the guard cell here. This one. So the guard cell here. Okay, this one. Guard cell, this one. But this is the stoma inside there. It's asking asking you about. Okay, the difference in stoma is between the two species. Can answer? Can answer? We have here more stomata in the Bovina mandra, but here there is no stomata. It's number 1. The distribution between two species, the differences. So you will describe from the like that. Okay, we have okay. We have here what is about the stomata, talking about in the day, different in the night. So in the lower, the upper, we describe it before. So when the car change to cobalt chloride or when the article 6, then this time you have another question here. If you remember the unit and the divisions, that's about unit and divisions here. We have in chapter 1, if you want to remember.
مستر، محدش سمع حاجة خالص. يعني أنا دلوقتي حضرتك بتشرح وأنا مش فاهمة أصلا حاجة، مش سامعة.
Okay, you hear me well now?
Oh yes, thank you.
Okay. So from when? From the beginning of the lesson, you don't hear the video?
No, we heard about 20 minutes.
You hear the video. So after that, you don't take part in the past paper, you don't know?
Yes. No, you started drawing.
I will send the past paper today.
Mister, you drew something. That's something you drew, we saw it. After that, everything is silent.
You after baby or after
Yes.
paper, after the first past paper,
After the past papers.
after the first past paper. So
Yes.
So I will begin from now. I will repeat in the time about immunity. Now you understand me? You hear me?
Yeah.
We are revising now chapter 10, chapter 11, chapter 12. Okay? Chapter 10, chapter 11, chapter 12. Beginning the bus paper, you don't know, I think, what?
Mister, you use past papers?
What?
You use past papers?
I use it now. The
We
book in
didn't
front of you, you see it well? Now the book in front of you, you see it well?
Yes, I see the book, but the
But
past papers.
before that, there is a lot of question about bus paper. You don't understand it or you don't see it?
We didn't see it.
You didn't see the bus paper at all?
Yes. Yes.
Okay. 1 minute, 15 minutes.
Thank.
That's all. So now I will revise in antibodies and immunity. What is the difference between active immunity and passive immunity? I want to Shahad. Shahad. What is active immunity? Shahad, you hear me? Okay. Okay, Balak, you hear me? What is active immunity?
Yes, I hear now.
Active immunity. Okay. So it is the defense against the pathogen by antibody production in the body. I want to go to another question about chapter gametic exchange in the human. We'll revive. Okay. About gametic exchange in the human, we have a lot of characteristics about the gametic exchange. I want someone to ask what are the characteristics of the respiratory receptions? What are the characteristics? I'm asking you. What are the characteristics of respiratory surfaces? Uno. Cannot to show this page. This page on 156. You are with me? It's about page 156. Okay? Or you should tell me or no? Characteristics of respiratory surfaces, you don't know? Anyone know? What are the characteristics of respiratory surfaces? The exchange of oxygen and the carbon dioxide across the body. There is a large surface area exposed to the gas. There is a distance across which diffusion has to take place of the smoke. There is a good blood supply, and there is a big differences in the concentration of the gas at 2 points about by the temperature. There is this is called characteristics of the respiratory surface. So now I will repeat the question. What are the characteristics of respiratory surface now? Who knows? You will take part of why? Grade 9. I want someone to take part with me now. Now we go to aerobic respiration. Aerobic respiration. Okay, Alala, Sally, you want something?
No, thank
Okay. We said respiration, we have two kinds of respiration. We have aerobic and anaerobic respiration. I want someone to give me what is the difference between aerobic and anaerobic. It's a simple question. You don't study why. Good mind, you don't study. You must study well. What is the difference between aerobic and anaerobic? Okay. What is the
Aerobic, aerobic respiration is the breakdown of glucose with oxygen, and anaerobic respiration is a breakdown of glucose without oxygen.
Thanks. Anaerobic. I want another one. Thanks, Anna. I want another one. I want who raised their hand? Kalala, Siri, and I want another one. What is anaerobic respiration? I wanted the equation of it, of the anaerobic. I said you must study chapter 10. Okay, Shahad. Shahad, you already finished?
So you need the definition of aerobic respiration?
Anaerobic respiration, yeah.
Mister, your voice is not clear.
My students, you are with me, you don't revise well, so to take bus paper, you must revise well. You don't revise well, I see the revise. Chapter 10, chapter 11, chapter 12. Okay, I return to you time another time. So I want to revise with you now chapter 11, 12, and chapter 2. We will revise now aerobic and anaerobic. Who can tell me, try to take part with me. Open your book to page 165. The next time, inshallah, on Sunday, okay, so what is aerobic? You don't study, just you need some moment work. What is aerobic respiration? Now, you said the term, okay. I want some student now. I will tell you. Just check. I want another one. What is aerobic? You must take part with me. Don't take part with the okay? I want another one. Where is the boy? Where is the boys? I want the boy to take part with me. That is vista. You don't take part, you don't study boys. Boys, you must study well. Okay? Okay, Allah. Thanks, Allah. What is anaerobic respiration?
Aerobic or anaerobic?
Anaerobic respiration.
Anaerobic respiration is the chemical reaction that in cells that break down nutrient molecules to release energy without using oxygen.
So thanks, Allah. Thanks. What about the equation of aerobic respiration? The equation of aerobic respiration. No, just I want just child and Allah. I want the other, only the boy, only the other student. Where is the other student? Muhammad Subhi, where are you? I'm the other student. Where is anaerobic respiration? We studied it. If you want to explain another time, okay, but you don't study what it is. Anaerobic respiration means that, as we said, the term for the chemical reaction in cells that break down nutrient through layers without urine. That is anaerobic respiration. Okay? But what about the equation of anaerobic respiration? It's in front of you now. We can say the equation of the anaerobic respiration. We can. What is the equation of anaerobic respiration? You remember all the students? Or no? What is the equation of anaerobic respiration? What is the equation? Just shut down the Just shut down the Salman, what is the equation? What is the equation of anaerobic respiration? What to do, Salman? What is this?
Salman.
Who is with me? Salman or what? Salman, you're with me? Okay, good to everyone. Anna, what is the equation? Anna, what is the equation of anaerobic respiration?
Glucose with the help of enzymes is broken down to alcohol plus carbon dioxide
Right.
and energy.
Bravo. And what is the energy here? 118 kilojoules. We explained in Anna. Okay, we explained aerobic and anaerobic in the last week. Okay, we explained it. Okay, you studied well. You studied well, Anna. Thanks, Anna. Thanks a lot. Okay. I want another student. Okay, what is the difference between aerobic and anaerobic? The difference between them in number one in definition, number two in equation. If you go here to aerobic respiration, go quickly with me to aerobic respiration in the equation. In the sample with glucose plus oxygen, that is the equation. The different enzymes will produce carbon dioxide plus water. But in aerobic respiration, this is the equation. But if you go quickly to page what? To page 167. So now that's it in aerobic respiration. What is anaerobic respiration? The term of the chemical reaction instead of breakdown nuclear but without using oxygen. Number two, okay, anaerobic that's an definition. Number two, okay, I will repeat anaerobic respiration without oxygen. Okay. In addition, we have the equation of anaerobic here. That is anaerobic respiration, glucose. In the case of enzymes, okay, produce alcohol, the carbon dioxide, okay, 118 kilojoules, that is the unit. So now I want anyone, okay, I'm explaining now. I want anyone to give me the difference between aerobic and anaerobic now. Okay, after that, we took the note paper, you must part with me. You can answer me just that. I want to avoid the word, the difference. I'm explaining now. What is the difference between aerobic and anaerobic? Okay, what is the difference between aerobic and anaerobic? Just alert. Read the boys. Read the boys. Read Muhammad Awad. Where? Are with me? Muhammad. You hear that? Muhammad Awad. You hear me well or no? You hear me well? You know the difference between aerobic and anaerobic? You must part with me. Huh? Abdullah. Read the other about. Okay, Ala, no problem. Ala, what is the difference? Okay.
The aerobic respiration is the chemical reaction in cells that use oxygen to break down nutrients to release energy.
Your voice,
Same,
please.
but without the use of oxygen.
The equation. Number two, the equation in aerobic respiration is
Glucose plus oxygen with the help of enzymes will produce carbon dioxide and water and energy. And the energy of released
I
by
want
the
another one.
respiration.
I want another one to give me thanks. Thanks. I want another one to give me. Okay, what is the difference between aerobic and anaerobic now? Thanks, sir. Thanks a lot. With the other students. I will repeat on that side. We said in anaerobic glucose, in the presence of enzymes, it will produce alcohol and carbon dioxide and 110 kilojoules. So now we want its campus paper. You see it well? Or no? About aerobic and anaerobic, which was and respiration. All right. This one. See this one the last time. This question between the respiration and transport. Describe the function of each of the following parts of the right atrium. No, that's an extension man. You ask about the center. That's an essential part. Describe the function of the. One of the questions here you know. I want someone to raise his hand. Okay, you raise your hand. Okay, who raise your hand now? You raise your hand. You are one. What do you want? Shahad. Shahad.
So here you need the function of the right atrium, right?
The bus paper in front of you is clear or no?
Yes.
Okay. What is the what is the function of right atrium?
So the right atrium receives the deoxygenated blood
Thanks.
from the body and through the vena cava and pumps it to the right ventricle to send it to the lungs oxygenated.
Thanks. Your name what? Shahad. Thanks, Shahad. Excellent. Okay, that is the gesture of the boys. The boys doesn't take part today at all. So you will see the bus paper well. What is the function of right ventricle? The blood separation between transport and respiration. What is the right ventricle? The separation between transport and respiration. Right ventricle contain oxygen. Just Ala. Another one. Just Ala and Shahid. Okay, Ala. Right ventricle.
Right ventricle will receive the blood coming from the right atrium.
Receive the blood, right ventricle from the right atrium, bravo, and go to the lung, okay?
And then it will.
Yeah, and we carry by pulmonary artery, we carry it to the lungs. Understand? So I'm very happy of the girls today, Ala and Elcha. But what about the boys? They don't take part at all. We have question here, please outline what is tricuspid valve. I want to take part. This question, inshallah, I will send it to you. Please answer it. I will send it to you today. This question and this question. Okay. Now, so this question is very important because you will see them maybe your hands. The tricuspid valve. What is the function of the tricuspid valve? Any valve. What is the function of the valve? You don't study ball at all. What is the valve? What is the function of the valve? No, I don't want to chat. They participate. What other student? Who is the other student? You must study well. Okay? Boys, what is respiration? You must study. The next time, chapter 10, chapter 11, chapter. You don't study. Okay, Muhammad Sobhi, thanks. Okay. Muhammad Sobhi, I hear you. Muhammad Sobhi? Anyone? What is respiration, Muhammad Sobhi? Muhammad Sobhi, Malak. I want to hear Malak, that's Muhammad Sobhi. I don't know why. Muhammad Sobhi, you don't want to take part? Can you revise or mic have a problem? Muhammad Sobhi, hear me, what is respiration? What is respiration, Muhammad Sobhi? What is respiration? What is the relation between them and the tricuspid valve? Okay. On chat, okay, chat. Respiration.
You want the
Respiration first, then what is the tricuspid valve? Yeah.
your voice is lagging, cannot hear anything.
Okay, no problem. What is the function of tricuspid valve in the tricuspid valve?
So the tricuspid valve, it closes to prevent blood from returning back again into the right atrium.
Thanks. Thank you. Thank you, sir. So today, my student, inshallah, a lot of students today, take part with me, chat. Thanks. Muhammad Sobhi, raise your hand, but I don't have any problem. From the Muhammad Sobhi, from the Ala, and I think I'm take part with me. But I wish for the student, please, I will, inshallah, the next time we'll solve chapter 10, chapter 11, chapter 4. Please concentrate and study well. You don't study at all. Okay. Thank you a lot for waiting. But this means I'm complaining of non-participation of some students. Why? I don't know why. You must participate. Okay? You must participate, my students, the next time. Thanks a lot. I will send to you, inshallah, today, the first paper, WhatsApp and from the site for both of you. You must answer it. Thanks. Assalamu alaikum.